Hi, my name is Danielle Tullman-Ercek, and I am a professor of chemical and biological engineering at Northwestern University, where I also co-direct the Center for Synthetic Biology. And today I'm going to talk about engineering a complex phenotype, how we've used high throughput approaches to develop a new protein biomanufacturing platform. So the motivation for our work is that recombinant proteins are a large and important market, whether you're talking about therapeutic proteins, industrial enzymes, protein materials, or other markets that aren't listed here. And to make all of these proteins, we can achieve really high titer production in bacteria. That gives us a really high quantity. The bacteria are easy to manipulate, they grow really quickly, they're not very expensive to grow. But unfortunately, the lab strains do not secrete functional non-native proteins efficiently, and so they are low quality. And so often you will find that those industries that I just mentioned will turn to other production hosts instead of bacteria to make their products. So we see this as primarily a failure in purification. When you are making protein in bacteria, you are necessarily going to have to deal with separating out your protein of interest from all of the other proteins that the cells are making intracellularly, or in some of the organisms that do have some robust secretion, they also secrete a lot of their own proteins in a way that we cannot completely eliminate. And so the goal when we started out on this project in my lab was to optimize protein production in bacteria for both quantity and quality. And specifically, we were aiming to produce over a gram per liter of protein, so a gram of protein per liter of cells that we were growing, with over 90% purity without doing any additional purification steps. And also more than 90% of those should be functional or active, in other words, properly folded. And so we want to do this again without those downstream purification steps, and so taking advantage of a secretion system within the bacteria itself. And we set these goals in part because we had been working with a system from Salmonella enterica, a particular cerevar from a mouse infecting strain known as Typhimurium. And it has this set of type 3 secretion systems, one of which is pretty well characterized, that had the potential to meet those tighter purity and cost objectives that I just mentioned for recombinant protein production in bacteria. And so these systems are really interesting. They are pretty large operons, so something like on the order of 23 to 25 proteins involved in making the structure itself, as structural proteins are involved with that process. And you can see it looks a lot like a needle and syringe, where the syringe is the green and pink part, and the needle is that basal body that's sitting across the two membranes, as well as the periplasm of the Salmonella, which is a gram-negative bacteria. And so the polypeptide actually comes from the cytoplasm and is secreted in one step all the way out of these cells. So we had been working with the system for some time in a variety of ways from the 2008 or 9 timeframe. And we had already seen that we could use known secretion tags. These are sequences, protein sequences or amino acid sequences that go at the N terminus of the protein of interest that you would want to secrete. So you can encode this genetically. And when you attach these secretion tags that are from natively secreted proteins onto whatever you want to secrete, we found that we could secrete a diverse set of proteins of interest. This was started in this work shown from 2009 and continued in my lab in some unpublished work. We actually tested about 80 different proteins that bacteria make, none of which are necessarily secreted natively. And about 80% of those 80 proteins were compatible with this secretion system. The system already was known to be able to secrete protein, but the titer is the really important part. And so with some initial work that we did in the 2010s, early 2010s, we were able to show that we could manipulate a little bit about the regulation of the system to get it to be active in all of the cells in the population and not just some as it was natively. And we were able to achieve over 100 mg per liter for many of the proteins that we tested. This is great. It's not quite the gram per liter that we were going for, and what I'll describe in the rest of this talk. And then the one other thing that we knew by around 2015 is that those proteins that we were secreting actually were folding correctly and were fairly high purity, around 80%, with some of the other proteins that were still being secreted from the native system at that time. And the correctly folded proteins included alkaline phosphatase and antibody fragments, which do have disulfide bonds. So even though we didn't have any chaperones in the media, they were able to fold properly. And so that was really exciting, and that's what led us to really begin wondering, how far could we push this system, and could it be used for commercializable applications? We were inspired, but we knew we had a lot of work ahead of us. And so on this slide, I'm showing that we need to do several things from that point to actually reach commercial relevance with this system. So the first is engineering the regulation of the system to control it. We're in a unique position with this system in Salmonella that it's not always present, and that's great. But also it has a very complicated network controlling its production, and the presence or absence of the apparatus itself is dependent on integrating a lot of signals. And so we need to overcome this regulatory control. And we did a little bit of that work early on. We found this master regulator, HIL-A, that by putting this on a plasmid and artificially expressing this master regulator, we're able to induce the system, but that's not the ideal method for industrial applications. We don't want to use expensive inducers, and we really wanted to avoid extra plasmids in the system as well. And so finding ways to control that was high on our list. We also wanted to make the secretion process more efficient. We were getting over 100 mg of protein per liter, but we were leaving quite a substantial amount of protein still behind in the cells unsecreted at the time points that we use. And so anywhere from 10 to 80%, depending on the protein. And of course, this is without optimization, but we wanted to see what controls that efficiency. And then finally, we needed to increase the titer to that 1 gram per liter mark that we had decided was a good benchmark for pursuing further if we could reach it. So here's the thing about these goals, as you may have surmised from the way I was talking about it just now, these were not easy phenotypes to engineer. It requires extensive genomic changes, regulatory cascade changes, and the phenotype in all cases is really how much protein is outside of the cell at the end. And we knew that for each goal here, success would likely require screening thousands to millions, billions, trillions, all the way up to maybe 10 to the 12 variations, basically as many variations as we could make in a test tube at the DNA level or genome level. And so when you compare that goal and the set of constraints that we predicted that we would have in achieving that goal, and then you compare that to what the gold standard is for assaying secretion as a phenotype, which is Western blotting, there's quite an incompatibility. And so I want to back up a second here and just talk about what Western blotting's process looks like. And so here I show we have our signal peptide, that's the secretion sequence tag that's at the N terminus of whichever protein that we would want to secrete. And we have a detection tag in purple at the very end of the protein of interest once it's produced off of this plasmid that I'm showing. And that detection tag is anything that you can detect with Western blotting. So if there's an antibody against it, that will work. And then we also have the chaperone shown in red. That's just to facilitate the secretion process. And it actually interacts with the signal peptide. So is not as important, but is important to represent here in the cartoon. So if we encode this cassette, this expression cassette on a plasmid, we will make our polypeptide with its signal sequence and its detection tag, and we will make the chaperone, which will help by binding to the secretion tag and guiding it to the needle or the secretion apparatus. In the meantime, if we were to take the cells and centrifuge them down so we have a pellet of just cells and look at what protein is present in them, we would see how much protein is being produced by doing a Western blot against the cells. Once we activate the secretion system and have it present, we would then be able to detect whether or not it's going out. So if we don't have the secretion system in place, or for some reason the protein is not being secreted because one of these pieces is not working properly, such as the chaperone of the signal peptide or the apparatus itself, if we do a Western blot on the supernatant, so the media from doing that centrifugation step where you remove the cells, then you wouldn't see a band on your Western blot. But if you are able to secrete the protein successfully through the apparatus, everything is going as planned, then you will see by assaying that supernatant, assaying the spent media, if you will see a band once again indicating that the protein was secreted. And so we can compare those if we're very careful about quantification, or we can compare how much is secreted versus what was in the entire culture. So supernatant and pellet alike combined without separating the two. And so that's the gold standard. That is not going to allow us to look at thousands or hundreds of thousands of samples quickly. You can look at something like 10 to 15 lanes per gel. Maybe if you have a larger gel setup, you can do more, but we're not talking about hundreds. We're still on the order of tens. And so we need new approaches for assessing secretion titer. Now, fortunately, protein-based assays abound, and some can and have been adapted for secreted product, and I want to focus on those today. So this slide shows 3 very commonly used protein assays in the protein engineering field, in a lot of fields, even if not trying to engineer the protein. The first is enzyme activity. And so an example would be alkaline phosphatase, which is an enzyme that cleaves phosphate groups off of small molecules and can be tied to the appearance or disappearance of a color based on which small molecule you've introduced with the phosphate. Maybe you cleave the phosphate and it leads to disappearance of color. You cleave the phosphate and color appears because of the product that remains after you take the phosphate off. Now, this assay is dependent on the enzyme being properly folded and is often a multi-step assay to carry out, but can be done in microtiter well plates, which substantially increases the throughput over a Western blot. And it does have tunable dynamic range depending on how much protein you have present. The second one is the growth assay. This is where really comes in handy for something like secretion. So if you have the presence of the protein there, it usually leads to growth, although it could prevent growth similarly to the enzymes. So if you secrete a toxic enzyme, you're enabling growth because the toxic enzyme isn't there. Or maybe you need an extracellular component to be present, and as long as that extracellular component is present and you have your protein of interest present, then you get growth on that particular media. Potential for really high throughput, but a really small dynamic range and really hard to tune these assays. And then finally, we have optical assays, and examples of those would be fluorescence or luminescence if you were to use GFP, green fluorescent protein, as a reporter, or a luciferase-based product that produces luminescence. And so these, you're almost always looking for appearance of signal when you have the presence of this reporter protein that's optical in some way. And it, again, depends on folding. I think in all cases that can be true. But optical assays do have an advantage in that they're usually one step, and so a little bit simpler. And again, that leads to medium throughput because you can do it in microtiter plates. But you do get that large dynamic range back even though you're at medium throughput. Okay. So I'm going to talk for a moment about what we've done with developing an assay based on enzyme activity. So this was some earlier work where we wanted to engineer the type 3 needle complex itself. That's the complex shown in green. And it's all made up of a single protein that multimerizes to form an annulus, so a hollow cylinder, as shown here, but it really does come all from a single monomer of alpha helical structure. And we knew that it was potentially important. It was implicated in other systems that were homologous to our type 3 secretion system as being important for the amount of activity of those secretion systems. And so we introduced mutations in every single position of the gene encoding this needle protein. It's called PRGI. So we introduced, instead of just doing an alanine scan where we introduce alanine at each position, we introduced each of the 20 amino acids using degenerate codons N and K at each position. And so the library size here is we're looking at the length of the PRGI protein in amino acids times 20, and that's how many we are screening. And so that is medium throughput because PRGI is not the largest gene. It's on the order of 100 amino acids in length. We then inserted this onto the genome, which is represented very poorly here as just a circular piece of DNA. And then we screened for secretion via alkaline phosphatase, which was appended to a secretion tag. So the library here is in the protein that makes up the apparatus rather than the protein that we're secreting. And then we used a microtiter assay for alkaline phosphatase activity to assess secretion. So this library in general yielded 40% of the variants were still functional. So we didn't actually completely destroy the activity of the apparatus in 40% of the library members. And all of these, remember, are just one amino acid away from the wild type sequence. And we actually had quite a few variants shown in green on this chart that gave us secretion activity that was much higher than our previous best variant that was taken by homology from the literature. And so this is a frequency, so it's a histogram. So the x-axis is really the only one that matters, and that's the relative secretion titer. So closest to the origin, we don't get any secretion. The wild type is the next hump, and then you can see quite a few variants do outperform the wild type significantly. So we could have looked at just those high secretion variants, and we did. But we also decided to look at all variants and take advantage of some next generation sequencing affordability that we have now. And so we sorted every single well from every plate. So this is, we maintained the plates that had the cells from the ones that we did the assays on. And we went back and split those out into low, medium, and high secretion pools and sequenced each of those pools. And this led to something that we'd call a secretion fitness landscape, where we're truly looking at the performance of every single point mutant in our library, every single library member, and whether it fell in the low, medium, or high category. And so that's shown by this heat map that you see on the right. And to orient you, the x-axis of this heat map is the amino acids. The y-axis is the number for the residue. So at the bottom closest to the origin of the graph would be the N-terminal amino acid. And as you go up the y-axis, you move to the C-terminus. And then the color is pink if we didn't associate it with a high secretion phenotype. And it's green if it did exhibit high secretion. And the amount of green, if it's medium secretion, more like wild type, it's a medium color of the green or teal color. And so it's too small to see here which amino acids correspond to which positions are really important. But hopefully you can see the trends around the first alpha helix of the protein is in the N-terminus, and you can see a lot of light green on the bottom half of this secretion fitness landscape, whereas you see a lot more pink on the top half, but also a lot more dark green, showing that we get the highest performance from mutations that are in the C-terminal helix, but also a lot more chance for making a mutation that would completely kill the activity for secretion. And so now I'm going to show a video to show how that mapped on to the actual structure of the needle. And this was fascinating to us because the heat map itself maybe is very informative if you spend a lot of time staring at it. But what was really striking is that when we watch this video, you will see that we're going to be pretending to be the polypeptide being secreted down the barrel or the center of this cylinder going out through the secretion apparatus. And it's going to look a little bit like a rifle barrel with that nice turn in it. And there's some grooves, major and minor grooves. And we thought, if anything, we've probably widened this tunnel with the mutations we made. And those C-terminal does face the interior. So we thought we're probably widening the tunnel. When in fact, as you'll see in the video in a second, the green that you're going to see there is associated with mutating small residues to be much larger and bulkier residues. So here we will watch the video. All right, so we're going down. Oh, I'm sorry, I used the wrong color coding on this. So here red is not good and blue is the one that is the mutations that we were able to make. So I'm going to show it one more time from the beginning. And so again, you can see that those blue residues, that's where we're mutating to fill in more of this cylinder. And it's pretty striking that there's so much blue inside here, which corresponds to the green on the previous slide. And so this has led to a lot of new activity in my lab, which I don't have time to go into today. But I do want to just take the last few minutes to talk about another assay that we developed to test all the hypotheses that came from that really interesting mapping of the heat map onto the structure. And with that, I'm going to talk about an optical assay that we developed. So as I mentioned, a fluorescent reporter seems like a more natural way to go. And you might have even been wondering why we used an enzymatic reporter in the prior study. The answer is that, yes, it would be great if we could secrete a fluorescent protein and then just use an optical property or even a luminescent protein, or one that confers luminescence. You'd spin down the supernatant and collect it and look at the fluorescence of that. And directly detect your fluorescence. It's one step. It would be really easy. Fast, easy, and low error, very quantitative. Unfortunately, green fluorescent protein and all its variants are not compatible with our secretion system. This is often a problem with secretion of green fluorescent protein. The chromophore prevents it from unfolding for transport across the membrane. And so we found from work by David Baker and his colleagues a monomeric fluorescence activated protein, or MFAP, and thought it might be a promising alternative to use for our secretion assays. And so in this assay that we've developed, oops, my animations are going haywire, and we see that we are able to detect fluorescence when we add substrate. It does require adding substrate to our media that contains, hopefully, a secreted MFAP protein if we have the secretion system present. We don't see any fluorescence without the secretion system present, and we don't see any fluorescence if we have the secretion system, but not that fluorescence activating protein. The secreted MFAP protein actually gives us a signal that's linear across 3 orders of magnitude, so tremendous dynamic range, regardless of the media that we use, and that's what this plot is showing. And we were able to use it to explore our N-terminal secretion tags in more detail. And so I'm just going to back up to talk a little bit more. I mentioned earlier there's a chaperone. It turns out that the secretion tag itself has a chaperone binding domain as well as a secretion signal to it. And we wanted to see what role each of those pieces played in conferring secretion or secretability to our proteins of interest. And so our goal for a model secretion tag would be the highest secretion titer possible and the highest secretion efficiency. And we thought these might be toggled differently by the signal versus the chaperone binding domain and chaperone. And we also wondered if there would be differences observed with different proteins of interest. So there are quite a variety of tags and chaperones to choose from, which we wanted to mix and match. And that leads to quite a combinatorial challenge that we used our MFAP assay to address. So the first thing we did was make sure that our MFAP assay using fluorescence actually matched the gold standard of the Western blot. And this plot here is showing that testing each of the secretion sequences found in nature in its normal form in Western blotting as well as in the fluorescence assay gives us essentially the same results when normalized to the secretion titer of SBTP, which was the primary sequence we had been using in the past. And then we did that mixing and matching with our MFAT protein of all the different secretion signals, all the different chaperone binding domains in every order possible, to give 42 different experiments. And we found indeed that we could assay this with MFAP, and that actually the very best one was a combination that was not a natural combination of chaperone, binding domain, and signal sequence. So that was really exciting that this particular A5 sequence is not one that's found in nature. And so then we thought, okay, let's take this further, because that took about less than a week, start to finish, to do that whole experiment on 42. But that's not really stretching out this assay too far. So we looked at all of the other advances that we had made that I described earlier in this talk. Methods of increasing the secretion titer, where we engineered the filament or needle, the PRGI. Removing the tip complex was some of our earlier work that I alluded to earlier. Changing the regulation of the system, using the HIL-A master regulator that I mentioned earlier on a plasmid, and in other ways where we manipulated the genome to control the levels of HIL-A using modifications to other HIL proteins. And then finally, we even made changes to the media, testing a lot of different media and isolating one in particular that we thought was really the best by far because of its ability to both lead to high cell growth and expression of our secretion system. And so we combined 10 different strains, 2 different methods for expressing the HIL-A, and 2 different media in the end for testing this MFAP assay. And we were able to show, again, a surprise, that our very best performing strain and plasmid and media combination was actually one that doesn't require any plasmid and takes advantage of all of the changes that we had made to the cell line itself. And that there were a couple of others that really do have high fluorescence as well, indicating very high secretion titer that you can see in the far right lane of these heat maps, where dark blue is how much protein is secreted as measured by that MFAP assay. And so we compared these results back to Western blotting on just those key, the key strain plasmid combinations. And so the one highlighted in red on the heat map there is shown on the far right on the bar graph. That's on the left on here. And so what we're seeing now with all these improvements additively is something on the order of 3 to 4-fold more titer. So we now have titers over 400 mg per liter. And we were able to do that once again in basically less than a week, all these experiments. So we are able to do dozens of experiments at a time with MFAP, but we think we can extend this to hundreds at a time. And then just to wrap up a little bit on something that I said was important to us early on, we wanted to show that this advances that we make with the secretion system aren't just going to apply to our reporter protein. So in this case, the fluorescent reporter, or in the prior example I gave, the enzyme reporter of alkaline phosphatase. And so here I'm showing that the results are upheld. We still get the 2 to 3-fold improvements that we were seeing when we use other reporter proteins that are actually models of human proteins that we might want to produce in this way. One is human intersectin, and the other one is human growth hormone. So that's the black and the gray bars, respectively. And so hopefully from this, I've given you some information on the various assays that you can use to look at protein, either expression, secretion, or presence, depending on what you're trying to assay. In my case, it was secretion, so presence in the supernatant was what we were looking for. And also that protein secretion machinery are really amenable to engineering via targeted library methods, and what was lacking was these high-throughput screens. And so we can use fluorescence or enzyme assays to take the place of Western blotting in the future. And I think this can extend not only to others studying type 3 secretion systems in various bacteria, but those studying the secretion systems in any organism should be able to apply these same medium throughput methods that far exceed the low throughput Western blotting. And they can be coupled, as we did, with comprehensive mutagenesis and next generation sequencing strategies. And so I'm really excited about that. I want to thank, with my few seconds, all the students shown in bold here that contributed to the work. I showed their pictures, and so they will know who they are, but Lisa Burdett really spearheaded a lot of the early work with Kevin Metcalf and Hanteng Wang on this, and more recently, Sam Leach and Julie Liang have done a lot of the later work that I showed you. And in fact, we spun out a company, Opera Bioscience, which I am required to disclose, that uses the advances that we made in the secretion system. All of this work was funded by a variety of sources, including but not limited to the National Science Foundation and national labs, as well as Northwestern University. My contact information, website, and Twitter handle are here if you would like to reach out with any other questions. I'm also available by email or in the chat. Thank you.